Alright, so in this video I'm going to show you how to get insights into AlphaFold predictions using the AlphaFold analysis plugin run from VMD2. And so I'm going to select a file, let's say this one. It's a small protein with 150 amino acids. And as you will see, we get a number of plots. But before inspecting them in detail, let's take a look to the structures themselves. And so here is the AlphaFold prediction, which I'm going to center. There it goes. And as you can see, we have two colors. So the orange stands for amino acids with a confidence score less than 0.5, which is very low. And in yellow, amino acids with a confidence score between 0.5 and 0.7, which is low. These are scores delivered by the pLDDT module of AlphaFold. pLDDT stands for the Predicted Local Distance Difference Test. This is a regressor which is learned during the training stage of AlphaFold, and again here it just delivers small numbers. And so the first question is the following. So even though we see two sharp colors, so is it the case that the individual amino acids have such a clear separation for their pLDDT scores? And so we'll answer this question in a minute. And the second question is about the overall quality of the structure, because if we look at the structure, in fact, it does not look that bad. So if we move around, so let's say we have this strand here, which enters a linker, and then a helix, and then the helix has a short linker to a strand again. And so this mingling of secondary structure elements is in fact what is known as an alpha/beta fold. And so overall, the fold does not look that bad, even though, as we have seen, AlphaFold is giving very low and low confidence scores. And so we would like to in fact refine this pLDDT assessment based on something encoding the overall fold. So before doing this, I need to introduce a piece of machinery, and so in order to do this, I'm going to make two VMD selections. So for the first one, I'm going to select one amino acid, let's say res ID 95, and so I'm going to display it using the van der Waals mode. And so now you see the atomic balls of amino acid number 95, and so I'm going to make another selection, and in fact I'm going to select all the C alpha carbons, so name CA, within 10 angstroms of this residue, so and within 10 of res ID 95. Okay, and so let me display them in CPK mode. It will be lighter, and using as a color the chemical element. And so you see here again residue 95 in van der Waals mode, and all the C alpha carbons which are within 10 angstroms. And so with this I can start discussing what we see in the GUI. And so we'll first take a look to this plot, the first one here, first row, and so first column. And so the blue curve is about the number of amino acids which have a specific number of neighbors. And as you can see, we have two essentially local maxima. We have one maximum around 10 to 12 number of neighbors, and one around in 18. So the first maximum corresponds to the disordered regions, for example, this long tail here, and the second one corresponds to the dense packing associated with secondary structural elements, namely helices and strands. And so this curve tells you at a glance, in fact, what is the composition of your structure in terms of secondary structure elements. So it's quite informative. And so we'll be using this in a minute to refine the LPDT score in terms of the overall fold. Now, let's switch again to the pLDDT analysis, and we are going to take a look to this plot here, which is second row, first column. So let me magnify. And so if we focus on the blue curve, so first of all, let's note that we have negated the pLDDT score. So minus 60, minus 65 is in fact the top pLDDT we have for an amino acid in this structure, and minus 95 is the worst. And so even though we had two colors in VMD's graphical engine, as you can see here on this curve, we see that we have three regions with coherent pLDDT values. And in fact, the calculation which is behind this plot is a so-called calculation of connected components upon inserting the amino acids by decreasing pLDDT. So it's a bit technical, but that's a nice way to characterize the coherence of amino acids along the sequence in terms of pLDDT scores. And again, instead of 2 or 3 or 4 sharp colors, we can read out from these curves the number of coherent regions. And next to it, we have another plot which illustrates the presence of 3 regions. They correspond to the 3 triangles which we have here. This is a so-called persistent diagram, but I'm not going to explain it in detail. So this is the first question I wanted to answer. A refinement of the AlphaFold regions regarding pLDDT values. For the second question, I want to provide insights onto the overall quality of the prediction. And so instead of looking at a curve like the one we just looked at, we are going to look at a curve with a score which we call abstract. And this score is delivered by a regressor which is trying to recognize good-looking residues, meaning residues whose 3D environment resembles what we have in a known fold. And so here we have negated the scores too, and as you can see, so we have excellent scores, almost 100, and so we have poor scores in the 50s, meaning that in the structure, in fact, we have a whole range of scores, yet, as you can see, we have essentially one local maximum, extended local maximum, instead of having 3 regions. And indeed, if we look at the scatter plot to compare the pLDDT score and the abstract score, so the pLDDT score is spanning the entire range from 25, very, very low, to 65, which is almost high. But our abstract score instead is only providing values which are quite high, above 60, right? And so you see that these two numbers are independent. And in fact, now, if we look at the coloring of the structure provided by our score, so which we have here, it turns out that, in fact, our scoring function does not really distinguish between the helices and the strands because, again, they correspond to patterns which are visible in other known folds, in particular the alpha/beta or alpha plus beta folds present in fold classifications. All right, so this is what I wanted to show you here. So at a glance, you get interesting plots providing novel pieces of information about the coherence of pLDDT values on the one hand, and also an overall assessment of the fold of the entire structure.